In this video we will discuss about how the late phase activation of M1 macrophages cause the lack of arginine for lymphomas, and L-arginine is one of the important amino acid for the survival of lymphomas or the tumors that are developed around this lymph node. So let us understand what they have presented in their results and what are their final conclusions. So in order to check whether inos is responsible, is also plays a role towards this tumor death or not, what it is, they check the inos knockout cells in two different conditions, that is RMA QA1 tumor cells and B16F10 tumor cells. So in the RMA QA1 tumor cells, what they have find out is that when the inos has been knocked out, they see less tumor survivals even when it is treated with the vaccine. That is there is no uh, mice that are able to survive. However, in the case of wild type, uh, in the case of wild type tumors that in which the inos is not been knocked out, what they see that around 6 out of 12 mice are able to survive. Overall, what they have find out is that in the case of inos knocked out, only 10% of the mice are able to survive, but in the case of wild type cells which vaccine treated, they see the 83% of the mice are able to survive. That results are shown in the panel B, where we can see that in the case of wild type with vaccine treated, only less percentage or 10% of the 83% of the mice are able to respond, whereas rest of them are not able to respond. But compared to the other cases, inos knocked out with the vaccine, only 10% of the mice are able to respond to the vaccine, whereas 90% of the mice not able to respond, and control case is expected to be non-responder, that is wild type untreated and inos knocked out untreated. However, the other different results they have observed when they use B16F10 xenograft in the mice. In this case, they see a when the inos knockout tumors are treated with the vaccine and bispecific antibodies, they see a increase in the survival rate, and it is comparable to the wild type with bispecific wild type cells treated with bispecific antibodies. This indicates that there is something, or in a RMA QA1, inos plays an important role towards this tumor. Inos does not plays that important role in the RMA QA1 xenograft, but it plays an important role. I have said it reverse. In RMA QA1, it seems like inos plays an important role towards tumor death or killing of the tumor, whereas in B16F10, that inos knockdown does not plays a important role towards tumor killing. And there is the TMA analysis of RMA-QA1, which shows that in the case of percentage live cells, in the CD8 plus T cells, they see that CD8 plus T cells are high in the case of this inos knocked out with vaccine treated, but higher than the CD8 plus T cells that are present in the wild type. But for the macrophages, they do not see any significant difference. That is, difference is not statistically significant. This indicates that the inos does not indirectly does not affect the macrophages, but inos might somehow playing a role of effector immune cells. So other things that they check, the cytokines that are anti-tumorigenic cytokines, they are actually upregulated in the inos that are treated with the vaccine. For example, this CD44A is upregulated compared to the wild type vaccine, and also CD49A is also upregulated, and other anti-tumorigenic cytokines are upregulated, but still it shows a less survival rate. So this could mean that inos is somehow plays a role of effector immune cells. And for the macrophages in this case also, what they have find out is that in the case of these inos with vaccine treated, they see a increase in the PD-L1 receptors, which is the which is the, you can say, marker for checkpoint immune therapies receptors. And there is also upregulation of this Ly6, which shows that macrophages are increased, and other factors such as MSC2 is also increased, which indicates that macrophages are also increased. So inos is does not play a role towards inos. One of the hypotheses that they developed is that inos might convert L-arginine to L-citrine, and after that they will produce more NO that can kill the tumor cells. And when they check the nitrile in the cell culture medium, they see that in the M1 stimulated cells, they see that more nitrile formations, or more nitrile formations compared to the unstimulated cells. However, when they check the cell death, they do not find any statistically significant cell death in the nitrile stimulated culture medium compared to the unstimulated. So this indicates that the hypothesis that NO formations would cause more tumor death is not that much correct, as the phagocytosis killings is more predominantly compared to this upregulations of this NO. Thereafter, they check the literature to find out that L-arginine is important amino acids for the survival of lymphomas, and this RMA-QA1 are the tumors derived from lymph nodes. Then they check the L-arginines content in the medium and percentage of Ly cells, and they are able to see there is a positive correlations, which is R square of 0.99. And when they see the similar relations for B16F10 tumor xenograft model mouse, they see a negative correlations. So this they see negative correlations, and it also has R square very low. This indicates that tumors. This results indicates that the tumors indicates that inos dependence on this tumor survival, or inos functions as a effector immune cells, depends on whether the tumors are from the lymphomas or near the lymph nodes, or from the other region. If they are from the lymph nodes, then inos plays a role such as immune effector cells, and if they are not from the lymph nodes, then it might not play a significant role in tumor killing. So this is what they have conclude from this experimental results. So I hope you understood a brief overview of what they have concluded and how they come up to that conclusions. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.